So we go ahead, and I'm just going to recap a little of the last video. We have this helicase, and helicase is basically opening up the genetic material. And by doing that, it's gone ahead and make an opening for the DNA to be separated. Remember those hydrogen bonds have now separated? We'll have an enzyme that'll come, and we'll have some little enzymes that sit on the outside. They're called single-stranded binding proteins, and they're just holding it open so it doesn't collapse on itself. The next thing that'll happen is we have this one little guy that comes. It's called ligase. Ligase will put this little piece of temporary genetic information on the piece of DNA, and it is done so this protein called polymerase basically goes ahead and starts to fill in those gaps. So what polymerase does, it's kind of like a photocopy machine. Every time it sees an A, it puts a T. Every time it puts a C, it puts a G. And it continues and continues. But the problem is that this can only go in one direction. It can only read this information in one direction. And it reads it from a 5' to 3' direction. So over here, we have our 5' to 3' direction too. And these are dictated because of the parent strand. So you'll see that the parent strand, this outer strand, went from 5' to 3'. So when it copies, the opposite is happening. So this is 5, this is 3, so it's copying this direction. And this one, because it's 3 to 5, it's copying in this direction. That's the hardest thing to remember. But what is going to be the thing that you kind of need to know is how the gaps get filled in. What are these enzymes doing? So you'll see that we'll continue on. So the polymerase will continue on, and this polymerase at the top, he can go ahead and continue in this direction. He's basically following the helicase. This guy is going the opposite direction of the helicase because of that 5' to 3' directionality. So what's going to happen is that as the helicase keeps continuing on its direction, and keep in mind the helicase is going in this direction, what's happening is this polymerase is starting to fill in the gap. He's lucky. He could just sort of follow the helicase, no interruptions. So he's just going to wait for it to open up and just keep following it and following it and following it, no problems. This guy, he can only start to do 5 to 3. So what will happen is he'll have to make another one over here and basically start to fill in these gaps. And eventually this will open up again. We'll have to do another one and another one. And that strand, or that type of recording, or that type of thing is called the lagging strand. So here, because we can continuously keep going, it's called the leading strand. And this one's going to make little sort of, tiny strands on the other side. It's called the lagging strand. You might read it or hear other videos online, and they're called Okazaki fragments. But I like to use the word lagging strand. Leading strand, lagging strand. This can go in one continuous piece. This is going to go in little sort of tiny spurts. And you can see that the polymerase is still going 5 to 3. It opens up a little more. The leading strand is going to still continue. He's lucky. He's just going to follow. But the lagging strand had another piece, so it has to make a little piece over here. And then remember how he made this one first. He made this one second. He made this one third. Eventually this will open up. He'll have to make another one. So he's just making short spurts. And that takes a while, and that's why it's called the lagging strand. And he'll continue and continue until eventually what will happen is an enzyme will come in. And this new enzyme basically has a specific name, and it's basically called endonuclease. And remember how anything with an ACE means that it's an enzyme? And exo means either outside or it could be remove. So if you ever see this word or you see a word like this and you just don't know what to do with it, basically go ahead and take the name apart and see what's going on. It's an enzyme that removes something. And what's it removing? Nucleotides. So we had these little starting off points. Remember that ligase, primase went ahead and added? And when it does that what ends up happening is these little pieces are temporary. And there's definitely a lot more on one side than the other, and they have to be removed. And by removing them, this enzyme exonuclease will remove that little piece, and then our next enzyme is going to come in, and our next enzyme is going to be, called ligase. It still has an ASE at the end, which means it's an enzyme, and this is going to basically, means sort of like glue, so it's gluing the pieces together. So we had a little gap, like here, and it basically filled in that little gap with nucleotides. We do these replication forks. Remember those forks were these openings? We do these replication forks all over the genetic material. And we do this for speed. If we only did it at one side and sort of went to the other end, it would take forever. But by having all these starting points, it makes it so it happens much quicker and faster, and eventually we can get this process finished fast. And also remember that we called initially one of the strands, that top strand, we had two parent strands, and they separated. So we had one parent strand separate, another parent strand separate because of that ligase. And then we had to fill in those gaps with the polymerase. So we were able to make two copies. We call one of those copies the original ones. We call that the parent strand, and the new strand that was made, we call that the daughter strand, because that one's the new strand. We're going to double check it for a little while just to make sure that everything's perfect. And once everything is perfect, we'll go ahead and just let the cell start using that genetic information. In our next video, we'll talk about how we go from DNA and actually make protein.